Welcome to the deep dive. Today we're tackling something that, well, it touches nearly everyone. How those little pills we take to fight infections, oral antibiotics, can have a surprisingly big impact on the bustling community of microbes living in our gut.
That's right. We often think of antibiotics as, precision tools against harmful bacteria, and they certainly can be life-saving, absolutely. But the science we've looked at reveals a much broader story, where these drugs can also affect the trillions of beneficial microorganisms that are just so crucial to our health.
Okay, let's unpack this then. This gut microbiome, it's more than just a bunch of random bacteria hanging out, right?
Yeah.
Our sources really stress its vital role.
Absolutely. Imagine it as a thriving inner ecosystem,
Right.
seriously complex.
Okay.
You've got trillions of microorganisms representing thousands of different types, each with important jobs. Our sources show they're involved in everything from digesting our food and making essential vitamins to educating our immune system and even defending us against invaders. What's truly remarkable, and the research really backs this up, is that your gut microbiome is as individual as your own personal signature.
Wow. So when we swallow an antibiotic, what happens to this intricate society? It's not like it's only targeting the bad guys, is it?
Precisely. No. The information we've gathered makes it clear that many oral antibiotics have a broad reach.
Okay.
They don't perfectly distinguish between the bacteria causing trouble and the beneficial residents of our gut.
Yeah.
This wide-ranging action is what leads to the collateral damage we need to understand better. So a key takeaway right off the bat is that antibiotics can significantly disrupt our internal microbial balance. A big disruption.
So the mission of this deep dive then is to really understand what goes on in our gut when we take oral antibiotics, both, in the short term and the long term. We also want to explore what influences these effects and maybe what we can do to help our gut get back on its feet afterwards.
Exactly. We're investigating the immediate shockwaves and, importantly, the lasting echoes these essential medicines have on our inner microbial world.
Okay, let's start with the immediate aftermath. What does our gut microbiome actually look like in the days and weeks right after we've finished a course of oral antibiotics?
The picture our sources paint is quite striking. One of the first and most significant things we see is a rapid decline in both the variety and the sheer number of microbes in the gut.
A real plunge, huh? How dramatic is this drop we're talking about?
Well, our sources point out that within just a few days of antibiotic use, the gut microbiome of a healthy individual can temporarily resemble that of someone critically ill in the ICU.
Wow, that's intense.
It really underscores how significant this acute disruption can be. And this loss of diversity, it's so important because it reduces what we call functional redundancy.
Functional redundancy. Okay, what does that mean exactly?
Think of it like having multiple types of bacteria that can do the same job. If you lose a bunch, there's less backup.
Got it.
And the whole system becomes more fragile, less ecologically stable.
That makes sense. It's like a diverse workforce. If a lot of specialists suddenly disappear, the whole operation, well, it struggles.
Exactly.
So what about the specific types of good bacteria? Which ones are most affected?
Several key groups often take a significant hit. Bifidobacterium, for instance, which is so crucial for healthy development in infants, is often reduced by common antibiotics like amoxicillin. We also see drops in lactobacillus and bacteria that produce essential short-chain fatty acids, or SCFAs.
SCFAs,
like Faecalibacterium prosnitzi, Roseburia, and members of the Lactospiraceae and Rhumatocaceae families. Even bacteroids, a very common gut bacteria, can be reduced by certain antibiotics, like clindamycin. So a key insight here is that antibiotics can deplete some of our most beneficial microbial allies.
And these aren't just any old microbes, are they? They have really important jobs.
Absolutely vital jobs. These bacteria are critical for producing those SCFAs like butyrate, which is a primary fuel source for the cells lining our colon.
Okay.
It helps maintain the gut barrier and plays a crucial role in regulating our immune system.
Right.
Our sources also highlight something quite surprising. Some antibiotics, classified as bacteriostatic, meaning they're supposed to just inhibit bacterial growth, not kill them, can actually have a bactericidal effect, meaning they do kill many of these beneficial commensal gut strains.
So they're stronger than advertised, in a way.
In terms of impact on the microbiome, yes. The effect can be more severe than we might initially assume based on just the classification.
That's quite a revelation. So if the good guys are weakened, who takes advantage? It sounds like a vacuum is created.
That's exactly what happens. This is when opportunistic pathogens can step in. Our sources explain that when the beneficial bacteria are reduced, it creates an ecological opening, allowing these potential troublemakers to multiply.
Okay.
The classic example, of course, is Clostridioides difficile, or C. difficile,
C. diff.
which can lead to antibiotic-associated diarrhea and sometimes even severe inflammation of the colon, colitis. So the disruption of the good bacteria can definitely pave the way for the bad.
C. diff is definitely something we hear about and want to avoid. Are there other opportunistic players that can cause problems after antibiotics?
Yes, our sources mention others like Klebsiella pneumonia, Staphylococcus aureus, and various Enterococcus species.
Mm-hmm.
We also often see a rise in families like Enterobacteriaceae and the phylum Proteobacteria, especially in infants. What's fascinating and kind of worrying is that some antibiotics can actually increase the availability of nutrients that these pathogens love.
Like food for the enemy?
Sort of, yeah.
Yeah.
Things like sialic acid and succinate become more available. There's even this idea of collateral resistance.
What's that?
It's where bacteria that happen to be resistant to the antibiotic might inadvertently protect other harmful bacteria in the gut, kind of shielding them.
Wow. Okay. So it sounds like the whole delicate balance is just thrown completely off, creating this window of opportunity for less desirable microbes to really thrive.
That's a good way to put it.
What about the metabolic activities, the jobs the microbiome does? Are those immediately affected too?
Absolutely. Our sources emphasize that rapid shifts in metabolic function can occur even before we see the full changes in the types of bacteria present. It's quite immediate.
Really? Even before the populations change?
Yes. We observe altered gene expression and protein activity within the microbial community almost right away. And crucially, there's a decline in the production of those beneficial SCFAs we discussed.
Right, because the producers are gone.
Exactly. Simply because the bacteria that make them are reduced in number.
And I think I recall reading something about bile acids being affected as well. That seems important.
Yes, that's a really critical point the research highlights. Antibiotics can interfere with how our gut normally processes bile acids. It often leads to a decrease in secondary bile acids. These are modified bile acids that normally help to keep C. difficile in check.
Okay.
And at the same time, we often see an increase in primary bile acids, which actually encourage C. difficile spores to germinate and start growing.
Oh, wow. So it's like a double whammy favoring C. diff.
Pretty much.
Yeah.
It's almost a perfect storm that helps explain why the risk of C. diff infection goes up so much after antibiotic use. We also see changes in how carbohydrates are metabolized, potentially leading to a buildup of sugars that pathogens can readily use as fuel.
So more food for the bad guys again.
Yep. And it's crucial to remember, as the studies point out, that these metabolic changes aren't just a local gut problem. They can have ripple effects, systemic effects throughout the body.
Okay, so that's the immediate upheaval in our gut. Quite dramatic. What about the longer term? Do things always just, snap back to normal?
Unfortunately, not always completely. Our sources highlight several lasting effects of antibiotic use. One major concern is that the gut can become a significant and long-lasting reservoir for antibiotic resistance.
Resistance, right. We hear a lot about that.
Yeah, so a key long-term consequence is the potential for increased resistance to future treatments, both for yourself and potentially spreading resistance more widely.
How does that actually happen? How does the gut become a reservoir?
Well, antibiotics act as a selection pressure. It's basic evolution, really.
Okay.
If any bacteria in your gut happen to have genes that make them resistant to that specific antibiotic, they're the ones that will survive and multiply while the others die off.
Makes sense.
The gut then essentially becomes a storage facility for these resistant bacteria. And the genes that make them resistant, these are called ARGs, antibiotic resistance genes.
And they just hang around.
They can, yes.
Yeah.
What's concerning is that these ARGs can persist in the gut for a long time, months or even years, making future infections potentially much harder to treat. And it's not just contained within a single type of bacteria. These resistance genes can actually jump from one bacterium to another.
They can share them.
Exactly. Through a process called horizontal gene transfer. So resistance can spread through the community, even to potential pathogens.
That's troubling.
And our sources also remind us, it's important context, that antibiotic use in agriculture plays a significant role in this broader resistance issue.
Right. It's not just medical use.
No. And even the transfer of resistant bacteria from mother to newborn during birth or shortly after is a concern.
So even if the initial infection is cleared, we might be left with a gut that's kind of primed for resistance down the line. What about the overall balance of the microbiome itself? Does it always fully recover its original state?
Our sources suggest that recovery is often slow, frequently incomplete, and highly variable from person to person. It's not a simple bounce back.
Okay.
While some populations of microbes might recover relatively quickly, the microbiome may settle into what's been termed an alternative stable state.
Alternative stable state.
Yeah.
So different from before.
Yes, potentially. This new state might have lower diversity overall, or it might function differently metabolically than it did before the antibiotics hit.
Mm.
It's important to realize that even if the overall number of different types of bacteria seems to return to previous levels, like the diversity score looks okay, it doesn't necessarily mean that all the key beneficial strains have come back.
Ah, so the key players might be missing.
Exactly. Or the full range of metabolic functions might not be restored. The concept of an altered self is even discussed in some literature.
Altered self.
Yeah, suggesting that repeated courses of antibiotics could lead to a sort of new normal, a baseline for our gut microbiome that's actually less than optimal.
So a crucial point then is that the long-term recovery can be incomplete and lead to a new, potentially less healthy baseline for our gut.
That's a really good summary of what the research suggests, yes.
And this altered state, this potentially less healthy state, can make us more vulnerable to other infections later on, right?
Precisely. The reduced colonization resistance we mentioned earlier, the gut's natural defense against invaders, means it's less able to fend off new threats.
Okay.
We see this very clearly with the high rates of recurrent C. difficile infection. The initial antibiotic treatment disrupts the gut, allowing C. diff to potentially overgrow, and then the resulting persistent imbalance makes it much easier for C. diff to come back again after treatment stops. It's a vicious cycle sometimes.
Yeah.
Our sources also point to increased risk of serious infections like sepsis and vulnerable populations like premature infants, and animal studies show a higher susceptibility to other gut pathogens after antibiotic exposure.
Okay, so beyond these sort of gut-specific issues, our sources suggest links between this long-term dysbiosis, this imbalance, and broader systemic health problems. This is where things get really, really interesting and maybe a bit concerning.
Yes, the connections to chronic diseases are a major area of ongoing research. It seems imbalances in the gut microbiome, especially when they occur early in life, are increasingly being linked to a higher risk of developing various non-communicable diseases later on.
Like what specifically? What's the evidence pointing towards?
Well, one prominent example is inflammatory bowel disease, or IBD Crohn's, and ulcerative colitis.
Okay.
Studies consistently show people with IBD have reduced microbial diversity and specific imbalances, like fewer firmicutes, think F. prosnitzii, and more proteobacteria.
And antibiotics play a role there.
It's looking like a potential risk factor. Early life exposure and repeated antibiotic use, particularly certain types like those targeting anaerobic bacteria and cephalosporins, are being investigated. The link isn't fully proven causality yet, but the association is there.
And it's not just problems directly in the gut, is it? I remember seeing allergies like asthma.
That's right. Asthma and other allergic conditions like atopic diseases. Early exposure to antibiotics has been linked in numerous studies to a higher incidence of these later in life.
Why would that be? What's the connection?
The thinking is that disrupting the developing gut microbiome during those critical early windows, when the immune system is also learning and maturing.
Right, the co-development.
Exactly. It can interfere with the establishment of proper immune tolerance. We see things like reduced levels of bifidobacterium or early colonization by certain other bacteria like Moraxella in kids who later develop allergies.
Interesting.
And broad-spectrum antibiotics seem to have a stronger association, as does antibiotic use by the mother during pregnancy. It all kind of ties into the hygiene hypothesis.
Ah, yes, the idea that we're too clean.
Sort of. That reduced exposure to a diverse range of microbes early in life might skew the immune system towards allergic or inflammatory responses. So early antibiotic use might be an unintended consequence contributing to this.
I was also really surprised to see metabolic disorders like obesity and type 2 diabetes on the list of potential long-term consequences.
Yes, this is another really active area of research, and the evidence is building.
Mm.
Early life antibiotic exposure has been associated in several large studies with an increased risk of becoming overweight or developing type 2 diabetes later.
How?
Yeah.
What are the proposed mechanisms there?
It's likely multifactorial. Proposed mechanisms involve changes in how efficiently we extract energy from our food, alterations in the production of those SEFAs, which influence metabolism, increased permeability of the gut lining, sometimes called leaky gut, which can lead to chronic low-grade inflammation throughout the body,
Okay.
and even direct effects on metabolic signaling pathways.
Yeah.
It's quite fascinating. Even very low doses of antibiotics given to farm animals have been shown to increase body fat, which is actually one of the early clues.
Wow.
And we often see altered ratios of major bacterial groups, like the Bacteroides to Firmicutes ratio, in individuals with these metabolic disorders, though the significance is still debated.
And the potential reach seems to extend even further, maybe influencing things like irritable bowel syndrome, IBS, and even neurocognitive conditions. That seems like a huge leap.
Well, the evidence is definitely more preliminary in these areas, but
Yeah.
yes, there are potential links being explored. For IBS, we know that small intestinal bacterial overgrowth, or SIBO, can sometimes be triggered by antibiotic-induced dysbiosis, and SIBO symptoms overlap a lot with IBS.
Right.
And the fact that an antibiotic, rifaximin, can actually be effective for certain types of IBS, the diarrhea-predominant type, strongly suggests a microbial component is involved.
Okay, that makes sense.
And then there's the whole microbiota gut-brain axis, this communication highway between our gut microbes and our brain.
Yeah, fascinating area.
It is. And disruptions in the gut microbiome, potentially triggered or worsened by antibiotics, could theoretically affect brain function, mood, and even behavior through this complex network. It's early days, but the potential is there.
And our sources also mentioned something about dose dependency, like more antibiotics equals more risk.
Yes. Some studies have pointed towards a dose-dependent relationship, particularly for some of these chronic conditions. It suggests that the cumulative effect of multiple courses of antibiotics over time could be significant. It's not just one course, perhaps, but the repeated hits.
This paints a really complex, intricate picture. So thinking about the antibiotics themselves, the specific type, the dosage, how long we take it, these are all obviously really important factors, right? They must influence the impact.
Absolutely. Our sources definitely delve into the various factors that modulate how antibiotics interact with our gut microbiome. The characteristics of the antibiotic itself are crucial, as you'd expect.
Like broad spectrum versus narrow spectrum.
Exactly. Broad-spectrum antibiotics, as the name suggests, target a wider range of bacteria, so they tend to be more disruptive. Some studies suggest they can affect up to 30% of the gut bacteria.
Wow, 30%.
Yeah. And they've been more strongly linked to things like asthma risk. Narrow-spectrum antibiotics are theoretically less disruptive because their targets are more specific.
But still have an impact.
Oh, yes. Their impact still depends heavily on which specific bacteria they target and how susceptible our beneficial commensal microbes happen to be to that particular drug. There's work on developing even narrower spectrum drugs, like ibizapolstat for C. diff, which seems to have less collateral damage.
Okay. And dose and duration. Higher and longer is worse.
Generally, yes. Higher doses usually lead to more significant disruption, and longer courses obviously give the antibiotics more time to exert their effects and can result in slower recovery. There's a known link between longer duration and increased C. diff risk, for example.
But it's not always that simple.
No, it's nuanced. Some research indicates that even relatively short courses of potent antibiotics can still cause significant and lasting changes. And sometimes other factors, like the specific antibiotic class or host factors, might actually be more influential than moderate differences in duration.
Right. And of course, whether the antibiotic is taken orally versus, say, IV, that must make a big difference to the gut.
Huge difference. Oral delivery means the drug passes directly through the gut in high concentrations, leading to a much more direct and significant impact on the gut microbiota compared to IV administration. Although 5 antibiotics can still affect the gut via excretion into bile, for example.
Okay, so that's the drug side. What about us, the hosts of these trillions of microbes? Do our own characteristics play a role in how our microbiome responds to antibiotics?
Definitely. Host-specific variables are just as important, if not more so in some cases. Age is a huge one.
Especially early life, right? You mentioned that window.
Exactly. Particularly during those critical early life periods, the first 1,000 days or so, from conception to age 2. This is an especially vulnerable window.
Why then specifically?
Because the microbiome is still developing, it's getting established, and the immune system is maturing right alongside it, learning from it.
Right, they develop together.
Yes. So antibiotic use during this time can have more profound and potentially longer-lasting effects on the microbiome's development, leading to delayed maturation, reduced bifidobacterium, increased proteobacteria, and that's strongly linked in epidemiological studies to the increased risk of those chronic diseases we discussed earlier, asthma, obesity, IBD.
And even things before birth matter, like maternal antibiotic.
Yes, absolutely. Factors like whether the mother took antibiotics during pregnancy, the mode of delivery at birth C-section versus vaginal, which affects initial colonization, and how the infant is fed breast milk versus formula, can all influence how a baby's microbiome develops and how it responds if antibiotics are needed later.
And I imagine our diet throughout life is a major player in all of this. It seems like it always comes back to diet.
It really does. Diet is arguably one of the most powerful regulators of our gut microbiome, day in and day out.
How does it influence the antibiotic impact?
Well, a diet rich in diverse plant-based foods, high in different types of fiber, generally supports a more robust, diverse, and resilient microbiome to begin with. And after antibiotics, such a diet really aids in recovery. It provides the necessary fuel prebiotics for beneficial bacteria like bifidobacteria and CFA producers to bounce back. It helps promote faster recovery and can even help reduce those antibiotic resistance genes.
And the opposite is true, too. A poor diet makes things worse.
Absolutely. A typical Western diet, often low in fiber, high in processed foods, sugar, unhealthy fats, tends to support a less diverse microbiome and can really hinder recovery after antibiotics. It might even make us more susceptible to opportunistic infections like C. diff.
Any specific foods or nutrients mentioned?
Our sources mention specific types of prebiotics like FOS, GOS, inulin found in things like onions, garlic, bananas, whole grains, also things like mucin glycans, and even xanthan gum in some studies. And interestingly, one animal study suggested that eating a low glycemic index diet alongside antibiotics helped attenuate some of the negative microbiome shifts.
Interesting. What about our genes? Does our DNA influence this?
It likely does. Our own genetic makeup probably plays a role in shaping our baseline microbiome composition, and potentially influences how our microbes, and we as the host, respond to antibiotic challenges. This is a really exciting area for future personalized medicine approaches, tailoring treatments based on genetics and microbiome.
Yeah, that makes sense. And finally, just the starting point matters, right? The health of your microbiome before you take the antibiotics.
Definitely. It stands to reason, and the evidence supports it. A more diverse and stable gut microbiome before antibiotic exposure generally shows more resilience. It tends to recover faster and perhaps more completely than a microbiome that was already depleted or unstable.
Okay, so we've looked at the immediate fallout, the potential long-term consequences, and all the factors influencing the impact. What does the actual road to recovery for our gut microbiome look like? Can it bounce back on its own? How long does it take?
Our sources suggest that there's a natural recovery process, but it's really important to have realistic expectations about the timeline and completeness.
Okay, so what's realistic?
Well, partial recovery of some bacterial groups might occur within, say, a month or two for many people after a typical course of antibiotics. But regaining the full spectrum of microbial diversity, getting back to pre-antibiotic levels, can often take 6 months or even longer, according to some studies.
6 months or more?
Yes. And as we discussed, some specific beneficial species might take years to recover to detectable levels, particularly after certain potent antibiotics like clindamycin studies show effects lasting up to 2 years. Or some species might even be permanently lost, just gone.
Permanently?
It seems possible, yes. Some studies haven't detected certain species even 180 days after broad-spectrum treatment. And those antibiotic resistance genes we talked about, they can also persist in the gut for a considerable amount of time, maybe 1 to 2 years or even longer after the antibiotic course finishes.
So recovery happens, but it's often slow, maybe incomplete.
Exactly, and not always a full return to the way things were before, right?
Right. You mentioned those alternative stable states.
That's a key point from the research. Often, the microbiome doesn't just snap back perfectly. It might reorganize and settle into one of these alternative stable states that could have lower overall diversity or function differently, metabolically speaking.
So even if the diversity numbers look better eventually,
it doesn't guarantee functional recovery. The specific beneficial strains that do important jobs or their full range of metabolic activities might not fully recover even if the general diversity index improves. And there's just huge variation in this recovery process from one person to another. It's definitely not a guarantee of a complete return to the pre-antibiotic state.
Okay, so given that recovery can be slow and incomplete, what steps can we actually take to help our gut microbiome recover? Are there specific strategies we can use to support this process?
Yes. Thankfully, there are things we can do. Our sources discuss several approaches, and probably the most foundational one, as we touched on, is diet.
Diet again. What kind of diet helps recovery?
Prioritizing a diverse, plant-rich diet that's high in various types of fiber is really crucial. This provides those prebiotic compounds.
like the food for the good guys.
Exactly.
Yeah.
They act as food specifically for our beneficial microbes, like bifidobacteria and lactobacilli, helping them repopulate and produce those beneficial SEFAs. Incorporating fermented foods like yogurt, kefir, sauerkraut, kimchi can also be helpful as they can introduce beneficial microbes and their metabolic products.
And things to avoid.
Well, minimizing repeated or prolonged antibiotic use whenever possible is obviously key. And generally being mindful of other stressors on our system, things like chronic stress, other medications like acid blockers, maybe environmental toxins. But diet really stands out in the research as a powerful modulator.
More powerful than other interventions sometimes.
Potentially, yes. Some experts argue it might be even more effective long-term than interventions like fecal microbiota transplantation if you don't also cultivate a supportive dietary environment. You need to provide the right conditions.
Right, you can't just put seeds in barren soil.
That's a great analogy. Think of the recovery process as ecological succession, like a forest regrowing after a fire. Diet provides the necessary foundation, the right soil conditions, for the desirable microbes to flourish and outcompete less desirable ones.
Okay, so diet is foundational. What about specific interventions like probiotics, prebiotics as supplements, and symbiotics? Do they have a role to play in recovery?
They potentially do, yes. Probiotics, those live microorganisms intended to provide health benefits, have probably the most evidence, particularly for helping to prevent antibiotic-associated diarrhea, or AAD.
Yeah.
Specific strains like the yeast Saccharomyces boulardii and the bacterium Lactobacillus rhamnosus GG, LGG, are well studied and show good results in meta-analyses for reducing AAD risk when taken alongside antibiotics.
So they help prevent the side effects. Do they help the original microbiome recover faster?
That's actually a more complex question, and the evidence is mixed. Some studies, somewhat controversially, suggested that taking certain multi-strain probiotics might actually temporarily slow down the return of the person's own native microbiome diversity compared to letting it recover naturally.
Will they slow it down?
That's what one prominent study suggested. But others have found no negative impact or even potential benefits for restoring certain functions. So it's still an active area of research. The mechanisms are complex competition, immune modulation, barrier enhancement. Timing might also matter.
Like when you take them?
Yeah, often recommended to start them when you start the antibiotics, continue for a bit after, and maybe take them a few hours apart from the antibiotic dose, if it's a bacterial probiotic, to get a chance to survive. Yeast-based ones like S. boulardii aren't affected by bacterial antibiotics, which might be an advantage there. Higher doses sometimes seem more effective too.
Okay. And prebiotics, the fiber supplements.
Prebiotics, those non-digestible carbohydrates that selectively feed our good bacteria, are definitely considered important for post-antibiotic recovery. They directly fuel the beneficial microbes we want to encourage.
So taking fiber supplements after antibiotics could be good.
It could be, yes. Things like FOS, GOS, inulin. They can help boost bifidobacteria and lactobacilli, increase SCFA production, and some research suggests they might even help reduce the persistence of those antibiotic resistance genes. Of course, getting them from whole foods is ideal.
Whole grains, fruits, veggies, legumes.
Exactly. But supplements can be an option. One note of caution might be when to emphasize them, maybe focus more on them after finishing the antibiotic course, as high fiber intake during treatment could potentially interfere with antibiotic absorption for some drugs, though evidence is limited.
Good point. And symbiotics, the combo.
Right. Symbiotics combine probiotics and prebiotics, aiming for a synergistic effect. The idea is the prebiotic enhances the survival and activity of the probiotic strain.
Makes sense. Do they work better?
They might be more effective than either alone in certain situations, like potentially for managing obesity or maybe reducing AAD. The research is still growing, but the concept is logical.
Okay. And then for the really tough cases, like recurrent C. diff, there's the more drastic option, fecal microbiota transplantation, or FMT.
Yes, FMT is a recognized therapy now, particularly for recurrent C. difficile infection where other treatments have failed.
How does it work again?
It involves transferring carefully screened fecal material from a healthy donor into the recipient's gut via colonoscopy, enema, or capsules. The goal is to basically do a hard reset, introduce a whole diverse, healthy microbial community to restore diversity and colonization resistance.
And it works well for C. diff.
Remarkably well. Cure rates are often cited in the 70–90% range for recurrent CDI, which is fantastic.
Wow.
So research is definitely exploring its potential for broader microbiome restoration after severe antibiotic disruption, not just for C. diff and for other conditions linked to dysbiosis, but there are challenges.
Like finding donors. Safety.
It's exactly. Rigorous donor screening is crucial to avoid transmitting pathogens. Standardization of the process is tricky, and we still need more data on long-term safety. There's also exciting research into using a person's own stool, collected before they got sick or took antibiotics, and transplanting that back afterwards. That's called autologous FMT.
Using your own healthy sample, that sounds safer.
It could be, yes. Takes away the donor risk. And interestingly, the recipient's diet after FMT seems to influence how well the transplant takes and functions. Diet matters even then. Some research also suggests maybe it's not just the whole bacteria, but maybe microbial products or even bacteriophages in the filtrate that contribute to the effect. Lots still to learn.
This has been a truly deep dive into a really complex and important topic. So just to kind of recap the main takeaways, antibiotics, while essential, have a significant and often lasting impact on our gut microbiome,
Definitely profound impacts.
with consequences that can ripple out, affecting not just our digestive health, but potentially our systemic well-being too. And the recovery process, it's intricate, it's variable, and it's influenced by a whole host of factors.
Yeah, exactly. And that really underscores the importance of using antibiotics judiciously, what our sources often refer to as microbiome stewardship. It's about protecting this vital inner ecosystem.
Right. Minimizing unnecessary antibiotic exposure whenever possible, both for our own health and to help combat the huge global challenge of antibiotic resistance.
Absolutely crucial.
And it's encouraging to know that research in this area is constantly advancing, isn't it? We're continually learning more about these complex interactions between drugs, microbes, and us.
Yes, and exploring really innovative new potential therapies. Things like microbiome-sparing antibiotics that are much more targeted, engineered probiotics designed for specific tasks, phage therapy using viruses that target bacteria, and eventually maybe truly personalized interventions based on an individual's unique microbiome profile. It's a very rapidly evolving field with a lot of promise.
So, for you, our listener, maybe this is a good time to reflect a little on the microbial legacy of any antibiotics you've taken in the past. And perhaps more importantly, consider how your current dietary and lifestyle choices might be supporting or maybe hindering the health and resilience of your own inner ecosystem for better long-term well-being.
Yeah, maybe it's worth consciously thinking about incorporating more of those prebiotic-rich foods, those diverse fibers, into your diet regularly. Or perhaps looking into the potential role of specific probiotics if you face antibiotic treatment in the future.
It definitely raises a really important question for all of us to consider. Given these profound and sometimes surprisingly long-lasting effects we discussed today, how can we all be more proactive in nurturing our gut microbiome? Not just thinking about it after a course of antibiotics, but really making it an ongoing part of our overall health strategy.
That's the key question, isn't it? How do we actively cultivate resilience in this vital internal organ? It's a fascinating and increasingly important area with real implications for all of our health, present and future.